students, and welcome in the online session of class 9th Science, chapter 5, the fundamental unit of life. Students, we have seen that a cell is of two types, a prokaryotic cell and a eukaryotic cell. In a prokaryotic cell, there is no well-defined nucleus. That means the nuclear membrane is absent. Whereas in case of eukaryotic cell, there is a well-defined nucleus present as well as the membrane bound cell organelles are also present in it. So now we have to discuss the main parts of the eukaryotic cell. So we have already discussed that there are three main parts of the cell membrane, nucleus and cytoplasm. We have already started with the cell membrane. Now today in this session we are going to discuss some more parts of the cell. So let us see what we are going to see today. We'll cover cell wall, its structure, functions, then the process of plasmolysis, stages of plasmolysis, deplasmolysis, nucleus, and then the cell organelle endoplasmic reticulum. So let's first start with the cell wall. As we have discussed that in an animal cell, the outermost boundary is the cell membrane. It protects the cell. Whereas in case of plants, the outermost layer is the cell wall. Okay, so the plant cells possess an additional layer outside the plasma membrane. Okay, means they have cell membrane also, but outside the cell membrane, they have a cell wall also. Now this cell wall is non-living, like cell membrane is living. Plasma membrane or the cell membrane is living, this we have discussed. But this cell wall which is present only in the plant cells is non-living and it is rigid also. Rigid also, that means it provides hardness to the plant cell. Then it is completely permeable. It allows all the types of substances to pass through it. So it is completely permeable. Whereas the plasma membrane is selectively permeable or semi-permeable. That means it allows the entry and exit of some selective substances. So let us now see the structure of the cell wall. Now you have to refer this diagram that this cell wall is made of an insoluble carbohydrate cellulose. This you have to learn that cell wall is made up of cellulose and cellulose is a carbohydrate. And why we are calling it as insoluble? Because this carbohydrate cellulose is insoluble in water. Okay, so it is insoluble in water. That's why we have written that cell wall is made of an insoluble carbohydrate cellulose. It has pores. Students, this cell wall has pores that allow the exchange of materials between the adjacent living cells. Okay, means the cell wall has the cell membrane below it. Okay, so it exchange the material through these pores and pass on the materials to the next living cells. Living cells means to the cell membrane. Okay, now you can see the structure here that these are the cells. Okay, now this here you can find out that this is the cell membrane. This brown color is the cell membrane. Okay, here this brown color, this is the cell membrane. Fine, and cell wall is this one. This gray colored one is the cell wall. So this is one cell. Suppose this is two cell. Fine, so it has cell membrane inside and then above it has a gray colored cell wall. Okay, now you can see here these thread like structures. Okay, these are the openings, these are the pores, and actually they are called the plasmodesmata. They are called plasmodesmata. So through the help of these plasmodesmata or through the help of these pores, the materials can be exchanged through the cell wall. Fine. Okay, so this is the structure of the cell wall, that it is, you have to remember that it is made up of cellulose. It has pores through which it can exchange the materials to the nearby living cells. Now let us see the functions of the cell wall. Now cell wall, as it is hard and rigid, so it maintains the shape of the cell. It gives a particular shape to the cell. Then it has protection function. It protects the cell from mechanical injury. If the plant cell gets any jerk or shock from outside, then the cell wall will protect the cell and its inner parts also. Then due to its hardness, it gives rigidity and turgidity to the plant. Turgidity means it keeps the plant straight and swollen. And due to its hardness, it provides rigidity also. Okay, means the plant cell is not flexible, it is rigid, okay, stiff. Then it provides mechanical support also. It gives support to the plant due to which the plants can stand straight. Okay, so it provides mechanical support against gravity. Now, being freely permeable. Now, cell wall is completely permeable, so it allows the materials to pass in and out of the cells. Fine. So, these are the five functions of the cell wall: that it maintains shape, it protects the cell from mechanical injury, it gives rigidity and turgidity to the plant, then it provides support to the plant, and because it is completely permeable, it allows the entry and exit of the materials through the cell through the pores. Now, this thing you have to remember that whenever the word protoplasm will be used, so protoplasm is equal to cytoplasm plus nucleus. Okay, like suppose this is a cell, so there is a nucleus at the center. Now, this is what? This is cell membrane. This is cell membrane, and here the liquid filled is the cytoplasm, right? So, cytoplasm plus this nucleus means altogether whatever is present inside the cell membrane, that everything is known as protoplasm, right? Cytoplasm plus nucleus together they are called the protoplasm. So you have to remember this word whenever it will be used, you have to just keep in mind, so it will be easy for you to understand. Now let's see the next slide. Now here sorry, see this diagram also. This is the structure of the cell wall. Okay, there are these yellow color which you are seeing here, they are the cellulose strands. Okay, and this towards lower side, this is the plasma membrane, then cellulose strands are there. Okay, so this upper one is the structure of the cell wall. Okay, you have not to go in that much detail about cell wall, you have to just remember what is it, what material it is made up of, and what are the functions. Then let us move on to the next concept regarding cell wall, that is plasmolysis, right? Plasmolysis. Lysis means breaking. Okay. Now what is breaking here? Plasma. Plasma means whatever fluid is present inside, that is protoplasm, right? Protoplasm, now only I have discussed. Nucleus plus cytoplasm is called protoplasm. So the breaking of the plasma or the protoplasm or the shrinking of the protoplasm. Okay, now how it happens? Now we have studied the three types of solutions in the last video session. One was isotonic solutions, then hypertonic solutions, and one was hypotonic, right? Hypotonic, hypo, and this one is hyper. So isotonic solution, hypertonic solutions, and hypotonic solution. Isotonic solutions means where the amount of or the quantity of solute and water is same. Hyper means solute is more, solute is more. Okay, this arrow showing means more, upward direction means more. And in hypo, the solute particles are less, right? So this was about these solutions. Now let us see, when the cell is kept in these solutions, what happens. So when the plant cell in hypotonic solution receives water, now if a plant cell is kept in hypotonic solution, hypotonic means solute is less. Solute is less, that means water is more, because solute and water both are opposite to each other. If solute is less, water will be definitely more. So if you keep a plant cell in a hypotonic solution, then water will enter the cell by the process of osmosis, right? Because in the plant cell there will be a semi-permeable membrane, cell membrane, which will allows the entry of water inside. So when the water comes inside, the cell do not burst, okay, as it is surrounded by the cell wall. There is a cell wall present around the plant cell, so the cell will not burst. The cell wall counters the turgor pressure by exerting wall pressure. Now in the plant cell, what is happening, you can see here that when water enters here, right, so what will happen, the cell will swell but it will not burst. Why? Because this cell wall, this cell wall is also exerting pressure towards the inner side. This liquid water is exerting pressure on the wall, and this wall is exerting the pressure towards the inner side. Okay, so due to which the cell will not burst. Now this pressure stops the gain of water by the plant cell. When cell wall exerts pressure on the liquid and liquid exerts pressure on the cell wall. So when both the pressures become equal, then water will stop coming inside the cell. Up to a limit water can enter the cell, but when cell wall starts exerting pressure on the liquid on water, then water will stop entering the plant cell. Okay, this is the condition when the cell is kept in hypotonic solution. But now if the plant cell is placed in hypertonic solution, now hypertonic solution means the solute is more. Solute is more, that means water is less. If outside water is less, that means water from the cell will come out, right? So if a plant cell is kept in hypertonic solution, the cytoplasm along with the cell membrane shrinks, right? So what will happen now? Water will come out. So if water will come out, then definitely the cell will shrink. Cell is shrinking, that means cell membrane is also shrinking along with the cytoplasm. Everything will shrink. Now in this diagram here you can see it starts shrinking. Okay, they start coming in the center, and the cell wall has its place because it is rigid, but the cell membrane and nucleus, they all get shrinked at one place in the center like this. So the vacuoles also shrink, but they remains enclosed in the cytoplasm. Vacuoles will shrink, whatever cell organelles are present, they all will shrink and come in the center. The process of shrinkage of the protoplast, right? Protoplast now means protoplasm here, means complete protoplasm. Along with the cell membrane, nucleus, cytoplasm, everything will get shrink. Okay, due to the exoosmosis, means water has come out. So this is exoosmosis. So due to exoosmosis, all the protoplasm will shrink, and this process is known as plasmolysis. This is called plasmolysis, right? Plasmolysis means complete protoplasm will shrink and come in the center, and this is because of exoosmosis when the plant cell is kept in an hypertonic solution. Fine. So I think this is clear to everyone that plasmolysis means breaking of the protoplast, shrinking of the protoplast. And this will happen when the cell is kept in hypertonic solution due to exoosmosis. Water will come out and all the protoplasm will shrink at the center, and the process is called plasmolysis. This is the diagram for it. This is the plasmolyzed cell. You can see here that cell wall is intact at the place, but cell membrane, nucleus, everything is shrinked at the center. So this process is called plasmolysis. This happens in the hypertonic solution. Remember children, such type of questions can be asked in exam. What happens when a plant cell is kept in hypertonic solution? What happens when a plant is kept in hypotonic solution? So you have to keep in mind all the conditions. Fine. Now let us move on to the next concept of deplasmolysis. Right, plasmolysis we have studied. Now the word is broken from here, right? Plasmolysis we have studied. That means the cell has shrinked now. Now deplasmolysis means breaking the plasmolysis. Breaking the plasmolysis, that means again the plant cell will swell. In plasmolysis, the plant cell was shrinked, but in deplasmolysis, again the plant cell will swell, and all the protoplasm, that means nucleus, cytoplasm, everything will come back to its original place. Okay, so let's see how it happens. So if the plasmolyzed plant cell, plasmolyzed means which has shrinked, if such type of plant cell is immediately placed in a hypotonic solution, right? Plasmolysis occurred in hypertonic and deplasmolysis hypotonic. So when the cell is shrinked, now we are keeping it in hypotonic. Hypotonic means solute is less and water is more. So when water is more outside, so water will enter the cell now. Water will enter the shrinked cell. Okay. So when the cell is kept in hypotonic, water enters the cell and the plasmolyzed cytoplasm again swell up. So you can see here this was plasmolysis, all the protoplast collected in the center, but this is deplasmolysis where the water is going inside and everything then swell up and come back to the place. Fine. So all the cytoplasm will swell up and come in contact with the cell wall. Again they will come and stick to the cell wall. Okay, so this is deplasmolysis, means opposite of plasmolysis. So what solution is needed, children, for deplasmolysis? It is hypotonic solution. When the cell is kept in hypotonic solution, water will go inside and again the cytoplasm will swell and come back to its original place. Okay, so this was the last topic regarding cell membrane and cell wall. Okay, outer boundaries of the cell. Cell wall, remember, is only present in the plant cell. Okay, now plant cell also has cell wall plus cell membrane is also present in plant cell. Don't get confused. In animal cell only cell membrane, but in plant cells both are present. Fine. Now the next topic is nucleus. Okay, we have done with the outer boundaries. Now we have come at the center of the cell, that is nucleus. This is the structure for nucleus, children. So you can see in the structure that nucleus is a spherical structure which is present in the center of the nucleus, and it has double membrane, right? You can see double membrane here. One is the outer membrane and one is the inner membrane, right? So two membranes are there. It is a double layered structure. Okay, so it is bounded by two nuclear membranes called the nuclear envelopes. You can use the word nuclear envelope also and nuclear membranes also, same thing. Now it is perforated. Perforated means it has pores, right? So you can see here on the diagram, you can see one pore here, right? There is one passage here, then there is one pore here, then there one pore this side, then here also there is one pore which is written also. It is a nuclear pore, right? It is a hole in the nuclear membrane, so it is called a nuclear pore. One is here. So this nuclear membrane is perforated. Students, be aware that use some scientific words, right? If you will use perforated word, it will be much better than writing that nuclear membrane has pores. So use specific words. Try to learn these words. Okay. So nuclear membrane is perforated. That means it has pores, which are called nuclear pores. Okay, with numerous pores for movement of materials. Now what is the function of these pores? That these pores help in the movement of materials in and out of the nucleus, right? Many substances can come. Now this is nucleus, and outside what is present, children? This is the cell, right? So this nucleus is in the center and outside is the cytoplasm. So molecules can go inside the nucleus from the cytoplasm, and molecules can come outside in the cytoplasm using these nuclear pore. That means movement of materials in between the nucleus and the cytoplasm can take place with the help of these nuclear pores, right? Now the outer membrane bear ribosomes. Now you can see here green colored ribosomes are present. Fine, these are the ribosomes. These small green colored spherical structures are present on the outer membrane of the nucleus, and then these ribosomes are connected with the endoplasmic reticulum. This structure which you are seeing, this is the endoplasmic reticulum. Okay, and because ribosomes are present on it, so it is rough endoplasmic reticulum. In short, endoplasmic reticulum is called ER. So instead of saying the complete word, we can use ER word, right? So when ribosomes are present on the ER, it is called a rough endoplasmic reticulum. And if ribosomes are not present, then it is smooth. Now, so this we are discussing the structure that rough endoplasmic reticulum and sometimes smooth ER are also attached on the outer membrane of the nucleus. Now, nucleus contains fluid material called nucleoplasm. Like in the cell, the liquid which is filled is called cytoplasm, right? This is cytoplasm. And in the nucleus, the liquid which is filled is called the nucleoplasm, right? What it is called, children? It is called nucleoplasm. The liquid which is filled, the fluid which is filled inside the nucleus is called the nucleoplasm. Now, chromatin fibers are present in non-dividing cells. You can see here some thread-like structures. These threads which are present here. Okay, they are called the chromatin fibers. What they are called? They are called the chromatin fibers. These chromatin fibers are only seen when the cell is not dividing. Non-dividing cells. These chromatin fibers are visible only in the non-dividing cells. But when the cell starts dividing, then these thread-like structures become dense, compact, and they take this type of shape, right? We have discussed I think not discussed till now. Okay. So these chromatin fibers take the shape of this structure like this. And when they take this shape, it is known as chromosome. This is the structure for chromosome. Okay. This is chromosomes. Chromosomes. And when it is in thread like form, it is called chromatin when the cell is not dividing. When the cell starts dividing, these chromatin fibers will take the shape of a flower like structure, and at this time they are called chromosomes. Fine, they are called chromosomes. So you can conclude that chromosomes are only visible when the cell divides. Okay, when the cell is dividing, then only you can see the chromosomes. Otherwise, in a non-dividing cell, chromatin threads can be seen. Okay, now these chromosomes are made up of DNA and genes. Okay, now DNA structure is like this structure is there. This is the structure for DNA, right? So suppose this was chromosome. So this DNA is present in the, this was the half structure of chromosome I have discussed. So this DNA, this chromosome is made up of DNA, and one part of DNA, like suppose this much, this is one unit from here to here. This one part of DNA is called gene. This is called gene. This small part is the hereditary material which passes from parents to the next generation. Okay. So what can we say? That chromosomes are made up of DNA. This is DNA. Chromosomes are made of DNA. Chromosomes are made of DNA, and DNA is made up of gene. Okay. One part of the DNA is called the gene. Okay, and this gene is actually the hereditary material which passes from the parents to the next generation. We look similar to our parents because we have got genes of our parents, right? Now let us see the next slide, that this is again the structure for nucleus. One rounded body is called nucleolus. Now you can see here that in the nucleus there is one round again spherical body is present, which is known as nucleolus. So every nucleus has a round spherical structure in the center or at the side of the nucleus, which is called nucleolus. Okay, this nucleolus is actually floating in the nucleoplasm. Nucleoplasm is the liquid which is filled in the nucleus, right? So these nucleolus, they are dense, dark, with granular structure. Okay, granules, you can see some particles inside the nucleolus, so it's a granular structure. And this nucleolus is rich in RNA. Okay, RNA. It is ribonucleic acid. RNA, ribonucleic acid. Now, let us discuss the functions of nucleus. So, this is the general structure for nucleus, that it has two layers, outer and inner. Then, in the center, there is a nucleolus. Thread-like structures, chromatins are present. When the cell divides, chromatin get changed to chromosomes. And on the outer side of the nucleus, you can see endoplasmic reticulum, which are generally rough endoplasmic reticulum because they have ribosomes present on it. Now let's discuss the functions. That nucleus controls the metabolic activities and help in growth of the cell. Whole cell is under the control of nucleus. So you can say that nucleus is the controller of the cell. It controls all the activities, metabolic reactions which are going on in the cell. They all are controlled by nucleus, right? Then next is it forms ribosomes. These round structures which you can see on the ER, they are actually formed in the nucleus. Then it regulates the cell cycle. Cell cycle means the cell division, then cell duplication. They all are under the control of the nucleus. Means in short, you can write that nucleus is the controller of the cell. Every activity performed in the cell is under the control of nucleus. Okay, then it contains genetic information. Genetic information means gene. I told you gene, right? Gene is the hereditary material which is present in the DNA. So it also has the genetic information in the form of genes located in the chromosomes. Okay, so this was all about nucleus. So there is a body present inside the nucleus called nucleolus. It has nuclear pores also. Liquid inside is nucleoplasm, then inner nuclear membrane and outer nuclear membrane. This is the chromatin network present. These are the ribosomes present on the outer surface. Okay, when the cell divides, you can see the chromosomes also along with, this is the chromocenter now. A chromosome started appearing in the form, right? You can see this form. So this is the general structure and functions of the nucleus. Now let's move on to the next topic, that is cytoplasm, right? We are talking about cytoplasm for a long time. Let us see what it is. So in the diagram here you can see that in the center this is nucleus, this is the cell membrane. This is nucleus and this is the cell membrane, and in between the nucleus and cell membrane you can see a jelly like substance present, which is shown here in blue color, which is known as cytoplasm. So it is the fluid which is present in the cell in between nucleus and cell membrane, or the plasma membrane, right? And this cytoplasm consists of many cell organelles. You can see here mitochondria, Golgi bodies, ribosomes, vacuoles, they all are floating in the cytoplasm. So this cytoplasm consists of many cell organelles in it. Okay, so let us see the composition of cytoplasm. Cytoplasm is mainly, what is composition means, what is actually present in the cytoplasm. So cytoplasm consists of cytosol. Cytosol means the liquid watery plant. We are saying that this is jelly like substance. Okay, so other than liquid part, some cell organelles are also present in it. So the liquid part only is called the cytosol. We are going to study about it in the next slide. So cytosol is the liquid part of the cytoplasm. Then in the cytoplasm you can see the cell organelles. I have named many of them now. So cell organelles are present, and then cell inclusions are also present. One by one we'll see what are they. These three we have to study: cytosol, cell organelles, and cell inclusions, right? And this you have to remember, that nucleus plus cytoplasm is called protoplasm. Now let us see first point, that is what is cytosol. So the liquid or the fluid part of cytoplasm, liquid or fluid part of cytoplasm is called cytosol. Because it is now liquid part, so it is generally transparent in color, no color to it. And this cytosol contain 90% of the water. Okay, it has cell organelles also, and it has cell inclusions also. Then this liquid part also contains some ions, molecules like sugars, minerals, amino acids, enzymes, vitamins, they all float in this cytoplasm. All these molecules are floating inside. Okay, it contains the waste material also. The waste which is produced in the cell is also floating in the cytoplasm, right? In the cytosol, which is the liquid part of cytoplasm actually. Now let us discuss the functions. That it is the site of metabolic process. Metabolic means all the reactions which occur in the cell, they are actually happening in the cytosol. Okay, then it is the storehouse for many raw materials, many proteins, amino acids, enzymes. You can see here all the molecules, they are present in the cytoplasm, and they all are the raw materials for the reactions. So all the raw material is present in the cytosol. Then it distributes the nutrients, enzymes in the cell. Any molecule, it has to travel from one place to another. Then who is providing the medium to travel? So it is the cytosol. Then it brings about exchange of materials between the cell organelles. If something has to be transported from ER to mitochondria, suppose, then who will help in this transportation? Naturally, cytosol, right? Like we travel with the help of water media or not? We with the help of boats, we can travel the water and move to the other place, right? So in the same way, like something has to be transported from ER to mitochondria, then the medium is cytosol of the cytoplasm. The molecule will travel through cytosol and will reach to the mitochondria. So it helps in the exchange of materials also. Okay. Now next is cell organelles. Cell organelles means the membrane bound subcellular components. Okay, they are membrane bound, means they all have their own membranes surrounding them, and they are present in the cytoplasm of the cell. So they are called membrane bound subcellular components. They are the subcomponents of the cell, as they are present inside the cell. So they are the subcomponents, parts of a cell, right? And they are called the cell organelles. These cell organelles have a definite shape. All of them you can see here, these are the cell organelles: plastids, Golgi body, cytoplasm, vacuoles, then nucleus, endoplasmic reticulum, lysosomes, mitochondria. They all are cell organelles, and they all have their definite shape, structure, and they all have their function also. Now, these include plasma membrane. Plasma membrane is also cell organelle, cell wall, nucleus, ER, lysosomes. They all are cell organelles because they are present inside the cell. Now, presence of cell organelles is one feature of eukaryotic cell. That means these cell organelles are only present in a eukaryotic cell. They are absent in case of prokaryotic cell. Okay. Now, next slide is regarding the first cell organelle, that is endoplasmic reticulum. Now onwards we have to study in short all the cell organelles which are present in the cell. So first we are starting with endoplasmic reticulum, which in short is known as ER. Okay, so you can see the structure here of ER. This is the nucleus, children. Now it's very clear in this diagram. You can see the nuclear pores also. This is the nuclear envelope, means the nuclear membrane, and then here you can see the rough endoplasmic reticulum, and these small dots which you are seeing on the surface, they are the ribosomes. And here you can see the smooth endoplasmic reticulum also. You can see this part here, there are no ribosomes, so it is smooth, and where ribosomes are present it is called rough. Okay, so this endoplasmic reticulum is a network of intracellular membrane bound tubes, right? Intracellular means these tubes are present inside the cell, and there is a network of these tubes. You can see here this one tube, this second tube, third tube, right? So it is a network of, this ER is a network of membrane bound tubes. All the tubes have their membranes, okay, layers, boundaries. Now this is young in cell division. This is young in cell division, that means it is seen majorly during the time of cell division. It is absent in prokaryotes. ER is absent in prokaryotic cell. Now ER is of two types. One is rough endoplasmic reticulum, I have told you already, and one is smooth endoplasmic reticulum. So rough ER is called RER and smooth endoplasmic reticulum is called SER, right? Now this rough, why it is rough? Because there are ribosomes present on it. Okay, so this has large number of ribosomes on it. It helps to synthesize and secrete protein. Now you have to remember, children, that this ribosome is the place where proteins are synthesized, where proteins are produced. Okay, so this is the importance of ribosome, that ribosome is the place where proteins are made. That's why this rough endoplasmic reticulum is a very important cell organelle, because proteins are made here. And SER means smooth one. Here ribosomes are absent, so it helps in the synthesis of fats. So here proteins cannot be made. So what the smooth ER makes? It makes the fats and the lipids. Okay, so rough is making proteins and smooth one is producing fats and lipids. Okay. Now, some of the proteins and lipids synthesized in ER are utilized in building cell membrane. Whatever proteins and lipids and fats are made in the endoplasmic reticulum is used in making the cell membrane. We all know that the structure of the cell membrane is double layered structure. Okay, it is made up of bilipid, means two layers of lipids, and in between protein molecules are also present. So this protein which is made in the ribosome is used by the cell for the making of the cell membrane. Okay. So this is all about ER. Let us see the functions. This is again rough structure of rough, and this is the structure of smooth endoplasmic reticulum. So it acts as a supporting framework. Okay, it helps as a supporting, it helps to support the cell because tube like structure, so it provides again rigidity, it's provide a framework like bones provide a framework to our body. In the same way ER provides framework for the cell. It helps in the transportation of materials can be transported through these tubes. Okay, and packaging and membrane formation, as proteins are formed here, so it helps in the membrane formation because proteins are needed in the membrane formation. Then RER, rough one, helps to increase the surface area to synthesize proteins. RER means rough, means ribosomes are present. So many these dot like ribosomes are present which secrete the proteins and they increase the surface area also, right? Then smooth helps to store ions, detoxification and formation of Golgi apparatus, lysosomes and vacuoles. Smooth endoplasmic reticulum helps in the formation of other cell organelles also, like Golgi apparatus, lysosomes, vacuoles, etc. Then ribosomes provide the space for protein synthesis. Okay, so therefore ribosomes are called the protein factories because they are making proteins. Fine. So these are some of the functions of ER. Now let's see the next cell organelle, that is ribosomes. So ribosomes are dense spherical granules. They are small spherical structures. They are dense and they are like particles, and the size is 150 to 250 angstroms in diameter. This is the size of ribosomes. Ribosomes were first discovered by Palade, name of the scientist. Palade is the name of the scientist who discovered ribosome in 1955. Now, they are very minute organelles, very small organelles without a membrane around them. Remember, children, that majority of the organelles, they are membrane bound. They have membranes, but ribosomes do not have a membrane. Okay. Now let's see the functions. That main function is they are the place where the proteins are synthesized. So they are called the protein factories. Okay. So you can see here the structure of ribosome in the cell. This is the cell structure where you can see these blue small structures. They are the ribosomes. Then this is the structure for ribosome. Okay, it has two parts. One is this small subunit and this is the large subunit of the ribosome. Its body is divided into two parts, a small subunit and a large subunit. Okay, and you can see here the amino acids are linked together. These amino acids are the units that form the proteins. Okay, proteins are made by amino acids. So you can see here that amino acids are sequentially lined up here, making the protein. Now this is protein synthesis going on. Okay, now so these cell organelles we completed today. So let us see the summary. What we did, we understood the concept of cell wall, then nucleus, endoplasmic reticulum, and finally the ribosomes. So this much we have covered today. I hope the topics are clear. If not, you do watch the video again and again. Still, you can ask your doubts anytime. Read your textbooks. If you don't have the textbooks, please download it from net and just go for the textbooks reading. Okay, and complete your notebooks as well. So we'll meet in the next session. Till then, everyone, take care, stay home, and stay safe.